Yeah.
Okay, we'll get started, everyone. So thank you for coming to today's TINS. My name is Alistair, I'm the chair, and I have Holly, who is our co-chair. So today we have Dr. Mike Garratt. He's an associate professor from the University of Otago in New Zealand. Mike's originally from the UK. He completed his postdoctoral work at the University of New South Wales here in Sydney, and then went to the US, the University of Michigan. His research explores how reproduction shapes life history traits, health and aging, primarily using mouse models. His current work focuses on how maternal diets impact metabolic health during pregnancy, particularly in the context of gestational diabetes and the long-term health consequences for both mothers and offspring. Thanks, Mike.
Thanks, Al. Hi, everybody. So today I want to talk to you about a series of experiments we've conducted where we've been effectively using mice, manipulating the food that mothers eat during reproduction, and look at the consequences of that for maternal metabolic health, offspring growth and development, and also offspring health in adulthood. So there's 3 experiments I'm going to go through today, and I'll explain each one as we get to it. So that kind of gives you an idea of kind of how we're going to go through the talk. So it's well established that maternal diet, so what a mother eats during pregnancy and lactation, is linked to changes in offspring development, in survival, and also the future health of offspring. So epidemiological studies have shown links to a number of different aspects of maternal diet and outcomes for offspring. So intake of fat, intake of sugar, intake of protein, high or low protein, and intake in total amount of energy and maternal obesity. And all of these factors have been linked to different aspects of offspring development. So this shows that maternal diet is linked to changes in offspring health. However, in human studies, it's kind of hard to define kind of causal links between an aspect of diet and outcomes, because there are often confounding factors in eating. Well, first of all, measuring dietary intake of different components is quite hard in humans. And then also they can be correlated with other factors. So socioeconomic factors might correlate with diet, for example. People have also used mouse models to understand how maternal diet influences offspring development and health. People have looked particularly in the context of both overnutrition and obesity, and undernutrition as well during specific reproductive periods. So people have frequently used high-fat diets to manipulate intake of energy and obesity, and have shown that feeding a mum a high-fat diet causes impaired offspring development and particularly impaired health later in life. People have also used low-protein diets to model undernutrition. That's been studied for maybe 30 years, where you give a mother a low-protein diet during pregnancy or during lactation, and you look at the outcomes for offspring, and that can have really profound effects. So these studies can provide direct links between maternal diet and offspring outcomes. However, there are often confounding factors when you compare just one diet to a control. So, for example, a high-fat diet contains high energy levels. It causes maternal obesity. So it could be any of these factors that are actually causing those outcomes. Similarly, in the context of low-protein diets, they've been studied for a long time. And people have found different results depending on the type of low-protein diet they use and whether it has, for example, high carbohydrates or high fat to kind of compensate for that low protein. So we know there are links between maternal diet and offspring development, but it's kind of hard to establish the specific components of diet that are driving that. Maternal diet is also linked to the health of the mother during reproduction, and particularly pregnancy-associated diseases like gestational diabetes mellitus. So gestational diabetes is a type of diabetes that arises specifically during pregnancy. So in pregnancy, in all pregnancies, the baby, and in particular the placenta, produce hormones that impair maternal insulin sensitivity. And this kind of is expected to free up more nutrients and glucose to be allocated to the baby. So that happens in all pregnancies. And the mother's pancreas adapts to those changes and increases production of insulin to kind of compensate for that. Now, in most pregnancies, maybe 90% of pregnancies, these effects effectively balance, and the mother is able to maintain the balance of her circulating glucose levels. But in the situation of gestational diabetes, this fails to balance, and a state of uncontrolled hypoglycemia occurs. So gestational diabetes, like this impaired glucose tolerance, typically resolves after birth, but it has a number of short and long-term health effects for the mother and offspring. So gestational diabetes is associated with macrosomia, so babies being born large, preterm birth, and also an increased risk of preeclampsia. Although that hypoglycemia resolves after birth, the mother is still left with an increased risk later in life of developing type 2 diabetes. Some studies have suggested a 7-fold increased risk of type 2 diabetes, and also an increased risk of cardiovascular disease. And also babies from mothers who've been exposed to gestational diabetes also have an increased risk of these metabolic diseases when they themselves become adults. So a number of different studies have linked what a mother is eating during pregnancy to gestational diabetes risk. And there have been a number of different studies that have looked at this and found links to a number of different components of diet, and particularly different macronutrients. So the food we eat is made up differentially of different amounts of macronutrients. So some foods are high in protein, some foods are high in fat, some foods are high in carbohydrate. Now, typically, we actually balance this as we go throughout our day in terms of the foods we choose, but people still vary in the amount of these macronutrients that they eat, and this can associate with disease risk. So a number of different studies have linked high protein intake, either early in pregnancy or pre-pregnancy, to an increased risk of gestational diabetes. However, other studies have linked high fat intake, which occurs concurrently with high energy intake, to an increased risk of gestational diabetes also. There's then also been another study that's linked gestational diabetes risk to high sugar intake. So we've got lots of evidence that maternal diet can influence gestational diabetes risk, but we still have these same problems in epidemiological studies where it's hard to target exactly what's actually driving these changes. So I just want to touch on this high protein effect for a moment in human studies, because this might be quite surprising for some people. So there have been a number of different studies that have shown this link between high protein intake, either in pregnancy or pre-pregnancy, and the relative risk of developing gestational diabetes. So this is from a meta-analysis that was published last year, where they were looking at the relationship between maternal intake of protein and the risk of gestational diabetes. This is a forest plot, which basically shows you the relative risk of developing gestational diabetes in mothers who are eating high protein intake versus low protein intake. So about 20% of daily energy intake coming from protein versus about 11% coming from protein. If the bar goes to the right, it means that there's an increased relative risk of developing gestational diabetes reported in this study in women who are eating high protein. And what you can see is the bar is going to the right in nearly every one of these examples. And they actually reported that there's about a double the increased risk of gestational diabetes in women who are eating high protein compared to low. This is like a dose response meta-analysis here showing the change at different levels of protein intake. And you can see that as women eat more protein intake, their relative risk of gestational diabetes increases. We've actually been redoing a meta-analysis on macronutrient intake and gestational diabetes risk, looking at all macronutrients. And I just want to touch on one of those points is in our meta-analysis we're doing, we've also separated effects of animal protein versus plant-based protein. So what you can see here is actually these effects in studies. So this is from a meta-analysis. Each dot basically is an effect size that's reported in a study. So the size of the dot indicates kind of the precision estimate of that. So larger dots mean it's kind of a more accurate assessment. And we found that this increased risk of gestational diabetes, particularly when you're rather than plant-based protein. So as women increase their consumption of animal protein, their relative risk of gestational diabetes increases. whereas you don't see this in the context of plant-based protein. However, it's also worth noting that in general, people get less of their protein from plant-based sources than animal sources. But it suggests that there may be some link to the source of protein, not just the total amount you eat. Yeah.
How do they account for additional animal protein in the plant graph and plant protein in the animal graph?
Andy, could we say that to the end?
Yeah.
Question. Thank you.
Okay, I might not be able to answer it anyway, so that's
all right.
Hopefully you've forgotten about that question for me. Okay, so we wanted to understand whether there was any evidence that the intake of specific macronutrients during pregnancy could drive changes in metabolic health in a kind of causal way. So what we wanted to do is apply this approach called nutritional geometry that has been sort of pioneered by Steve Simpson and the rest of the lab here at the University of Sydney, to try to isolate the effects of protein versus carbohydrate versus fat on driving metabolic changes and changes in offspring development during pregnancy. So Steve and the lab have used a number of different approaches to manipulate the proportion of macronutrients in diet and look at outcomes, particularly using mice as a model. And one of the set of diets they've developed for use in mouse experiments is an isocaloric set of diets where they've got 10 different diets. They're isocaloric, so they have the same amount of energy per gram of protein per gram of food, but they differ systematically in the amount of energy they get from protein, carbohydrate, and fat. So you can see diets 1 and 2 here have 7% of their energy coming from protein. This is a really low amount of energy coming from protein. It's similar to those low protein studies that have been studied in the context of undernutrition. Then goes all the way up to a high level of protein, so 50% of the energy coming from protein, which is a really high level of protein in the diet. It has been used previously. The animals are still viable, but it's a high level of protein. Within this, we can then systematically vary the amount of energy that's coming from carbohydrate and fat. So these diets are presented as a proportion of the amount of energy you're getting from each macronutrient. And you can actually plot them on this graph here, which is a right-angled mixture triangle. So all the diets are made up of different proportions of these 3 macronutrients, so we can see them on this space here. So this is protein on the x-axis here, carbohydrate, the amount of carbohydrate on the y-axis, and then these diagonal lines represent the amount of energy that's coming from fat. So this dot here, for example, shows you diet 1, which is 7% protein, 33%, sorry, I can't see that dot very well, 33% carbohydrate and 60% fat. So you can see this line here is 60% fat, this is 40% fat, this is 20%. We can then go up and see on this space the other diet that is made up of 7% protein. You can see this here, it's got 7% protein, but now we've changed the proportion of carbohydrate and fat. So it's got 78% carbohydrate and 15% fat. We can then move down along this fat rail here. So we've got three diets that have 15% fat, and we can see diet 9 here. Again, it has 15% fat, but it's got 42% of the energy coming from protein and 43% coming from carbohydrate. And I drew those dots on so that they're approximate on there. So does that make sense to everybody? Mm-hmm. So we can put our mice on these diets and we can assess what it does to the animals, and we can look at a particular phenotype. So for example, we might be looking at body weight. And then we can look at how these diets influence body weight. Body weight in this hypothetical situation is our dependent variable. We then, in terms of data analysis, conduct mixture modeling, where we include all of these macronutrient proportions as predictors. And we fit a series of linear, nonlinear, and interactive models, including these variables. And then we see which model best fits the data using sort of standard model selection criteria, so on the basis of the AIC value. And we also test whether any of these models indeed are a better predictor than just a null model, so where none of these factors are included. For the best fitting model, we can then make a response surface. We can think about this as a heat map and plot this across that right-angled mixture triangle. It can show us what our particular trait of interest is doing. So in this particular example here, we can see that this trait is influenced almost exclusively by the amount of protein in the diet. So as the animals are eating, have got more protein, so as we go along the x-axis, the value is increasing. So red means a high value, blue means a low value. You can see that this trait is influenced by protein, but it's not influenced by changing the amount of carbohydrate and fat in the diet. So if the lines are going down vertically, it means that the trait is influenced by protein. If they're horizontal, it means the trait is influenced by carbohydrate. And if they fit across these fat lines, it means it's influenced by fat. Now, often we find interactive effects of macronutrients or effects of more than one macronutrient. So in this hypothetical situation here, in fact, this is a real plot I'll show you in a minute, but I want you to understand it first. You can see that we've got an effect of protein and carbohydrate. So as we go along the x-axis, you can see the color is increasing. So we can see it's positively influenced by protein. As we go up the y-axis, we can see it's decreasing. So it's negatively influenced by the amount of carbohydrate. But when we go along this diagonal axis, you can see it's not changing. So if you go for a given level of given protein carbohydrate ratio, the value isn't changing. So it's influenced by protein and carbohydrate, but not fat. Cool. So I'll talk you through the first couple of those graphs as we see them. Okay, so the first experiment that we conducted, we wanted to understand how maternal macronutrient intake during pregnancy influences glucose tolerance and offspring development. So what we did, we took mice, we put them on these 10 different diets for a week prior to being able to mate, to allow them to habituate to the diets. Half of the animals were then allowed to mate, and we allowed them to maintain their pregnancies until late pregnancy, so gestational day 17.5. Mice will normally give birth on day 20, day 21. And then we had another set of control animals that weren't allowed to mate and were just maintained as virgins. We sampled these animals for body weight changes, glucose tolerance, circulating insulin levels, changes at the level of the pancreas, fetal viability and growth, and also changes in placenta size and morphology. So these results, there's a preprint that covers the results of the first two experiments. So if you just Google that title, you'll be able to find it. It's been reviewed once and it's just been resubmitted. So hopefully it'll be published in the next couple of months. So the most striking result that we got in this experiment was the finding that high protein intake, specifically during pregnancy, impairs glucose tolerance in mice. So this figure on the left-hand side is a glucose tolerance curve. So basically we take the mice, we give them a bolus of glucose, and then we look at how that glucose is taken up from their bloodstream. So we serially sample their blood. What you can see is blood glucose levels increase after you give them this bolus and then decrease afterwards. And it's animals who are pregnant and on high protein diets where the blood levels go up the highest and stay up the longest. So you can see here in this plot, just for simplicity, I've split the diets into the low protein and the high protein. And you can see in the control virgin animals, there's no real effect of protein on glucose tolerance when they've been on these diets for just a couple of weeks. But for the pregnant animals, they're really sensitive to this effect of protein. So they have increased glucose levels on a high protein diet. So we can calculate the area under the curve of these graphs, and that gives us effectively a readout of their glucose tolerance. So a high area under the curve indicates impaired glucose tolerance. And I'm plotting that here in this figure on the right-hand side for the different levels of protein in the diet. You can see for the control animals, their glucose tolerance isn't really affected by protein at this time point. But in the animals who are pregnant, this is increasing. So the more protein they're getting in their diet, the more impaired their glucose tolerance. We can then create this response surface from the best fitting model. And it shows that in this experiment, the animals were influenced by protein and also to a certain degree carbohydrate. So here is the right-angled mixture triangle showing you where those different diets are. And then here is our response surface for the pregnant animals. You can see as you go across the x-axis, so you can imagine looking as you go across these diets here, there's an increased area under the curve. So high protein intake impairs glucose tolerance, gives a greater area under the curve. and more carbohydrate decreases this, but the strongest effect is for protein. We can compare this response surface to the surface we see in the control animals. We're using the same effective, kind of coloring, the same scale here. And you can see that in the control animals, we're really not getting very much of an effective diet on glucose tolerance at this time point. But in the pregnant animals, we're seeing this real sensitivity to the effects of diet on glucose tolerance. So we also measured fasting insulin levels in these animals at the end of the experiment. And here I'm presenting the data on an individual level where we're looking at fasting insulin levels in relation to the amount of protein each animal was eating at late pregnancy. So each dot represents a value for an individual animal, and it's basically looking at the amount of food that they ate times by the macronutrient composition of their diet. Again, you see very little effect in the virgin animals of eating protein, whereas in the pregnant animals, the more protein they're eating, the higher insulin levels that they have. Usually when we see increased insulin levels, it's indicative of impaired insulin resistance. So, and indeed, there's an interaction in our best fitting model between reproductive status and protein intake. So, at gestational day 17.5, we've looked at the pancreases. That's also in the paper. There's a strong effect of pregnancy on changes in the pancreas in terms of the level of the beta cells, but the effect of diet is quite minor on that. So going on to looking at the offspring, so looking at the fetuses at gestational day 17.5, we found that while high protein diets impair maternal glucose tolerance, they increase fetal viability. So when we dissected the animals, we looked at the number of dead fetuses and reabsorptions we could find in the uterine horns. And here, looking at each individual pregnant animal, we can see this negative relationship between protein consumed and the number of this measure of mortality. So animals who are eating the highest amount of protein had the fewer dead fetuses and reabsorptions. Or we can think about this in the other way. Animals who weren't getting very much protein had really high fetal mortality. Here we're expressing that information the other way. We're looking at the rate of fetal viability, which is kind of the more classic way you present this in reproductive biology. Red indicates very high viability, so most of the fetuses are viable. Blue indicates low viability. What you can see is this nonlinear effect of protein. So particularly on low protein diets, there's low fetal viability. This then increases until a maximum of when animals are eating about 30% of their energy from protein. So there seems to be a trade-off between maternal glucose tolerance and fetal viability. So when we dissected the animals, we also took the placentas. We want to look at the relative size of the placenta and basic placental morphology. We found no effect of the diets on total placenta size, or indeed the size of the fetuses, or the ratio between fetal size and placenta size, which is a marker of placental efficiency. However, we saw really dramatic effects on the actual placental morphology, in particular the relative size of the labyrinthine zone of the placenta, which is involved in nutrient exchange. That's actually where the fetal tissues and maternal tissues meet, and the junctional zone, which does various different things, including producing placental hormones. What we see when we look at the relative size of these areas is that as mothers are eating more protein, their junctional zone increases, but their labyrinthine zone decreases. So as you eat more protein, your labyrinthine zone, your junctional zone increases, but the junctional zone increases, but the labyrinthine zone decreases. I get to this point where I'm saying so many different kind of terms that I start getting confused, but hopefully you can follow me. So the easiest thing is to show you the figures. So these are actually the placentas from that study, and we're looking at the amount of protein and carbohydrate at each level of fat content. So here you can see the low protein diet. You can see there's a large labyrinthine zone, small junctional zone, and as we increase the amount of protein and decrease the amount of carbs, the labyrinthine zone gets smaller, the junctional zone gets bigger. And we see this at each level of the fat content. So there's a really dramatic change in the morphology of the placenta. And I think in part what's going on is the placenta is adapting to those changes. You know, we're not seeing a change in the placental efficiency when we look at it in a crude way, but the placenta has this ability to adapt to the macronutrients it's exposed to. So we can look at those surface plots here. Here is actually that plot I showed you at the start, that hypothetical example of a phenotype exclusively influenced by protein. We can see that that is the case in the case of the junctional zone. So as mums are exposed to more protein, their junctional zone gets larger, but their labyrinthine zone gets smaller. And we've also got this effect of carbohydrate as well. So the labyrinthine zone is reduced by high protein intake and increased by carbohydrates. So, in addition to doing that first study, once we've done that first study and saw these quite dramatic effects on fetal survival and placental function, we also wanted to look at the long-term consequences of what macronutrients, the macronutrients mums are eating during reproduction and the effects on offspring growth during lactation and the long-term consequences for the mother. So in terms of lactation, that's actually the time when mothers have to, particularly in mice, really have to invest a lot of energy in their offspring. So a female mouse eats 4 times more food during lactation than they would in the virgin state. And the mother undergoes major adaptations to be able to do that. In the context of maternal metabolic health, we were also interested in this interaction with lactation. While gestational diabetes is this type of diabetes which you see during pregnancy, if a mother is able to engage effectively in lactation, then some of the long-term risks of gestational diabetes can be decreased. So lactation is assumed to be beneficial for metabolic health, and that might offset some of these effects that we see in terms of diet effects during pregnancy. So what we did is we took the mice, we put them on these diets exactly the same as before. We allowed the female to mate. We allowed her to give birth. She stayed on these diets during lactation. We then weaned the offspring at 21 days of age. We measured offspring development during this lactation period. And then we left the mother for another 6 weeks and metabolically phenotyped her for things like glucose, circulating insulin levels, body composition at 9 weeks post-birth. So quite an extended period after they'd weaned their offspring. So this is the effect of these diets on pup growth. Apology, the figures are slightly blurry, but what you can appreciate is that we're seeing some really strong effects of these diets on pup size and weight across the lactational period. So this blue line here shows you the growth of the pups on the 7% diets at 5 days of age, 10 days of age, 15 days of age, and 20 days of age. What you can see is that they're really, really stunted, to the extent that they're actually half the size of the other pups when they're weaned. We actually didn't see an effect on pup mortality. The mothers look after these pups, but they're just really, really small. So they're really, really stunted in their growth. If we look at these lines carefully, so each line is a different protein composition of the diet, we can also see that we're seeing non-linear effects of protein on pup growth. So here's the 50% diet here. And you can see that this is also quite stunted, not to the same degree as the 7% diet, but pup growth is actually maximized on the 30% protein diet. Here we've got our response surface for average pup weight. Again, apologies that it's blurry, but hopefully what you can appreciate, so this is average pup weight at weaning. What you can see is that there's one specific diet that seems to maximally optimize pup growth and pup size by weaning. And that's the diet that has 30% protein, 30% fat, and 40% carbohydrate. If you move away from this target, if you have more or less protein, more or less fat and carbohydrate, then offspring size at weaning is decreased. Okay, so we also looked at the weight of our females across the experiment. So this is the weight of the control females, actually from the time that the reproductive females gave birth to the end of the experiment. What you can see is that these diets are isocaloric, so they have similar amounts of energy. The mice can eat different amounts of food, so there are some slight changes in energy intake. But what you can appreciate is there aren't major changes in the body weight according to these diets. However, when we look at our reproductive females, we can see really major changes in their body weight according to their diet from birth up to the end of the experiment. So what you can see for our reproductive females is on most of the diets, they really gain weight between birth and weaning at the end of the experiment. So actually there's about an 8 to 10 gram difference in the weight of our reproductive females compared to our control females by weaning. And this basically represents lean mass. So we actually looked at their body composition, their livers get bigger, their hearts, their kidneys get bigger, all to be able to absorb more nutrients and transfer them to their offspring. However, the 7% protein females actually lost weight over lactation. So they lost weight, they didn't put on the lean mass of the other groups. And then although these effects kind of diminished after weaning, they remained lighter. And if we look at their fat mass at the end of the experiment, these animals have much less fat than all the other groups. So actually, in a way, it's this low protein diet actually exposes costs of reproduction in these animals. When we put them on a low protein diet for the mothers, it seems to be more costly in terms of utilization of their energy reserves. So at the end of the experiment, we also looked at their glucose tolerance. We did the same glucose tolerance tests. Now, at this time point, the control animals had been on these diets for 4 times longer than they had been in that first experiment. And work by people here at the University of Sydney have shown that if you put mice on a high-protein diet for an extended period of time, this can lead to impaired glucose tolerance. And that's actually what we find in our control animals at this time point. So you can see there's this predominant effect of protein on glucose tolerance in our control animals. So our control animals, after 13 weeks on these diets, are showing impaired glucose tolerance. When we look at our reproductive animals, we actually find that all of the animals, irrespective of their diets they've been eating, have better glucose tolerance than the controls. So whereas pregnancy is this period where there's this enhanced sensitivity to the effects of diets on glucose tolerance, post-lactation, all of our females show a resilience to the effects of these diets on glucose tolerance. So the effects are effectively switched, which we hypothesize is the consequences of lactation. So we know that lactation can be beneficial for controlling glucose. It causes changes in the pancreas that means that you can release more insulin and also have increased insulin sensitivity. Okay, so the final experiment I want to talk you through was a long-term experiment where we wanted to look at how maternal diet, specifically during pregnancy, influences offspring health in adulthood. And this was really driven by those remarkable findings we found for the placenta and indeed for fetal viability, where we see that the placenta changes really dramatically during pregnancy. What are the consequences of this and those maternal diet effects in pregnancy for the long-term health of offspring? So in this third experiment, we took our mice, we put them onto these diets, we allowed them to mate, we left them on these diets until birth, and then at birth, we switched all the mice and put them all onto a standard diet on the day that the offspring were born. And we also adjusted the litter size so that the litter size was similar in all the groups. I think it was 5 to 6 pups in each litter. So we normalized the litter size, and that stayed effectively constant across from birth to weaning. So here, different to that last experiment, mothers are on the diets during pregnancy, but they're exposed to a standard diet over lactation. What I will highlight, though, is there could be carryover effects in terms of exposure to the diets during pregnancy on the lactational investment of the mothers. We wean the offspring, and then we assess their growth and body composition over this period, and then we've done a number of different assays on the animals at 100 days post-birth, so when they're fully unambiguously adults. So we've looked at their body weight, body composition, glucose tolerance, and we're doing a number of other follow-up studies on these animals now. So we did echocardiography, looking at cardiac fibrosis, cardiac hypertrophy, and changes at the level of the beta cell. Partly this is driven by previous assessments looking at effects of low protein versus control. These traits have been studied frequently in the 2 diet contexts, but as I highlight, there are other confounding factors, and we don't really know how these macronutrients can each individually and interactively influence these traits when we look at them across a scale. So the first thing I'll highlight is that we looked at the body weight of the pups at weaning, and we do see an effect of the diet that the mother ate in pregnancy on the body weight of our offspring at weaning, although it looks different to that previous plot where we actually left the mothers on the diets over the lactation period. What we can see is that we see, in particular, low body weights in the mice who are on the 7% protein, low-fat diets. So here is the response surface here. And here I'm actually plotting the raw data. So each error bar shows you an individual group and their weaning weight. You can see that the animals who are on the low protein, low fat diets weigh much less than the other groups. In addition, there's this effect of fat, in particular, if the mother was eating a higher fat level than 15%. So higher protein and fat increase the weaning weight of our mice. And the most dramatic effect was seen in this low protein, low fat group. So we assessed their body weight changes. So I'll just point out that this effect is exactly the same in males and females. So this, I think, is the surface plot for males, but the female one looks identical at this time point. So if we then look at the body weight of our animals when they're adults, when they're 100 days of age, this is the body weights of males, we can still see this negative effect of the low protein, low fat diets in terms of total adult body weight. So you can see these animals still weigh less, but we also see a negative effect in terms of total body weight for mice that are on high protein diets or the highest protein diets. So we're basically seeing a bimodal, non-linear effect of protein on body weight. And we're also seeing an effect of fat across those median protein levels, such that more fat intake also increases offspring body weight. What I'll say when we focus on this group here, which have this suppressed body weight, I haven't got time to show you it, but this is mainly an effect of lean mass. So if we plot the lean mass data, and I can show, if someone wants to see this, I can show you at the end. This group has the lowest lean mass of any other group. So basically, these animals were never able to catch up. They started lighter at weaning and they were never able to catch up. These animals were a normal weight at weaning, but something stopped them from being able to grow effectively from weaning to adulthood. When we look at the female body weights, actually in our tests, we found that none of the macronutrients actually predicted body weight in females. So in our female mice in adulthood, their total body weight was not impacted by the macronutrients that mothers ate during pregnancy. I'm still showing it, so this is like the next best fitting model. And what I want to highlight here is that the females are all lighter at this time point. So they all weigh about 23, 24 grams, whereas the males all weigh around 30 grams. So what I think is going on is that the males are sensitive to these diets because they show male-specific growth from weaning up until adulthood. They have an increased growth rate, put on more lean mass over the puberty period, and this means that they're more sensitive to those effects of diet as they occurred during pregnancy. So although we saw no effect on total body weight in our female mice, when we looked at their adiposity, so their percentage adiposity in adulthood, we did see a strong effect of the diets on the adiposity of female mice. So although their total weight was not impacted by maternal macronutrients, their adiposity was. So you can see that female mice who were exposed to high protein, low carbohydrate diets during pregnancy actually have the greatest proportion of fat mass. And this actually fits with some studies in humans which have associated with high protein intake during pregnancy to increased BMI in offspring. When we look at the males, we also saw an effect on adiposity. And you can see the lines here are pretty much horizontal. So this indicates an effect of carbohydrates on adiposity in the males. What I would point out is that this data is kind of complicated by the fact that these animals have really reduced lean mass as well. So their increased percentage adiposity could partly be a consequence of them having a low lean mass. And there's no real way we can kind of separate that. So the last figure that I'll show you relates to the glucose tolerance of these animals at the end of the experiment. So at the end of the experiment, we conducted glucose tolerance tests on these animals. Here I'm showing you the surface plot for males. So again, I'm plotting area under the curve. So a more red value indicates impaired glucose tolerance. And again, we see this bimodal effect of maternal protein intake on offspring glucose tolerance. So offspring from mothers who ate low protein diets or high protein diets had impaired glucose tolerance in comparison to animals on the medium diets. Just for reference, it's kind of hard to plot it when you've got these kind of non-linear effects, but here you can see the glucose tolerance curves for animals on the 7% protein diets, 21% protein diet, which is normal rodent chow, and the 50% protein diets. And you can see that it's high protein and low protein that both cause impaired glucose tolerance in offspring. We see the same effect in females, although it's less weak. So these macronutrients are still predicting female glucose tolerance, although the effects are less strong than as we see in males. Okay, so those are all the results I'm going to go through today. So in terms of our key findings, well, I think one of the key findings is that high protein intake during pregnancy impairs maternal glucose tolerance in mice. So we know that causally, and it's also consistent with epidemiological associations in humans as well. So there does seem to be some evidence that high protein intake during pregnancy can impair glucose tolerance. We've also found that maternal protein intake can impact offspring metabolic health also. So in terms of future questions that we'd like to explore, well, I'd really like to understand the specific amino acids that are driving that effect in mothers in terms of impaired glucose tolerance during pregnancy. It's been predicted in the context of human studies that high protein intake, in particular high animal protein intake, raises circulating levels of branched chain amino acids. This causes reduced insulin sensitivity in tissues, causing that impaired glucose tolerance. We can potentially take that apart using diets in mice that vary in specific amino acids. We're also following up on that finding in terms of offspring metabolic health, trying to understand how maternal diet influences offspring glucose tolerance. It's known, it's really well established that the low protein diet findings are linked to impaired development of the pancreas during specific windows during pregnancy. We're interested in finding out, is the same thing occurring with the high protein diets, or is there some other mechanism going on that's driving those effects? So how might these results relate and fit with dietary guidance in humans in the context of pregnancy? So in terms of dietary recommendations for food intake during pregnancy, I'm showing you on the left-hand side here a chart from the New Zealand Ministry of Health that shows different recommendations for the amount of different types of food you should eat for different sexes, age classes, and reproductive status. Now, this is kind of somewhat similar to the recommendations in Australia, but I couldn't find kind of a pretty plot to put in the diagram. In New Zealand, they're recommending that women eat, comparing reproductive age women to pregnant women, eating similar levels of vegetables, similar levels of fruit, more grain, and more, substantially more of foods that contain protein in them. So more legumes, nuts, seeds, fish, seafood, poultry, red meat with fat removed. In fact, if we looked at this scientifically, which we're probably not supposed to be, they're suggesting we eat 30% more of these during pregnancy. What about in the context of gestational diabetes? How do our results fit with nutritional approaches to respond to a diagnosis of gestational diabetes? Well, in response to gestational diabetes diagnosis, diet changes are kind of the first line of defense. And the guidelines that are used in response to this are largely the same as those recommended for type 2 diabetes. So it's recommended that women eat more or eat higher quality nutrient-dense carbohydrates rather than eating simple sugars, that they eat more unsaturated fats relative to saturated fats. But in none of the guidelines do they really talk about protein. And in fact, in response to these guidelines, typically what we see is that women actually eat more protein in response to a gestational diabetes diagnosis. Intervention studies where they've used diet to try to look at whether treatment of gestational diabetes with diet reduces some of those disease incidences like macrosomia, preterm birth. When people have done meta-analyses looking at how effective these intervention studies are, they've largely failed to show benefits of specific diet guidelines. So, as a consequence of these various results, I think a kind of key question is, should protein intake be moderated to some degree in pregnancy? Well, Number one thing is we do really need to ensure that appropriate nutrients are provided for fetal development. You wouldn't want anyone to suddenly start eating low protein during pregnancy, because protein, indeed, as we've shown, is really, really important for fetal development. However, in the current time, high protein is the thing. You know, everywhere you see various diets, various guidelines telling you to eat more protein and that it's going to provide health benefits. But in the context of pregnancy, particularly if you're already eating high protein, eating even more might have a negative effect. And indeed, there have been studies that have looked at drinking protein shakes and the consequences for gestational diabetes, protein supplementation, and have also seen increased risk. So it may be that protein levels could be tailored to specific intake levels. So if you're already eating high protein and you're aware of that, you wouldn't want to be eating even more protein. Another possibility is actually to promote plant-based proteins versus animal-based proteins. As we saw in that meta-analysis, we don't seem to see the same links between plant-based protein and animal-based protein. There's still a number of different questions that we need to answer, though, to actually understand how this is working in the context of humans, and if indeed it is. So we'd actually like to apply continuous glucose monitors to assess the relationship between diet pre-pregnancy and particularly early in pregnancy with continuous glucose levels. So is it a relationship to GDN diagnosis, or can you see it when you look at glucose levels in terms of a 24-hour period of glucose levels? Do we see a continuous relationship between protein intake early in pregnancy and glucose levels? And also, are there differences in the response to protein in a chronic context and an acute context? So we know that in the short term, eating protein can cause increased insulin secretion. So when you change to a higher protein diet when you're diagnosed with gestational diabetes, there might be short-term benefits in terms of changes in insulin secretion. And we really need to understand that. And there are potentially tools that we could do that. Okay, thanks everyone. I'd just like to, in particular, acknowledge the whole lab who basically ran these experiments as a team. So we all chip in and run these experiments. Faye has led the first two studies, Faye Morland, and Cam almost single-handedly has done that offspring experiment, which happened. I think the first offspring were born on Christmas Day, and the last offspring died on Easter. So he ran that almost single-handedly and was in the mouse facility every day. Thanks, everyone.
Okay. So questions?
Yeah, thanks for that, Mike. Questions already. Well, I think you have one.
Thank you so much and congratulations for such a great work. Very interesting. I wanted to understand if you have tried to look into the inflammatory status or inflammation immune components in mothers that were on high protein diet, because inflammation tends to reduce the thickness of labyrinth in placenta. It's a protective mechanism, but that hinders the passage of nutrient eventually. So that could be.
So inflammation, systemic inflammation?
Systemic inflammation, inflammatory cytokines, cells that are highly inflammatory and less regulatory in nature.
No, we haven't thought about that, but I would really like to do it. And
simply looking at inflammatory cytokines in your placental samples would be one.
So looking at it in the placenta, like looking at it in the actual placenta. Yeah, I mean, I've had so many ideas about what things to look at in the context of placentas. I would really love to do that. Yeah. So if you think that could be a possible mechanism, linking them, then yeah, I will.
It's like protection, but in a way, cause of sort of harming. Another small question. Have you looked into the composition of breast milk in women who were fed high protein diet? Does the breast milk composition macronutrient changes?
I'm sure it definitely does. We haven't done it because it's kind of like, we need a whole nother cohort of animals to do it. It would be quite a big experiment. 2 of the 3 reviewers asked us or suggested to do that, but we haven't been able to do it. I think they're going to be okay about it. So yeah, that is almost certainly a mechanism that we're seeing the changes in growth during lactation. Similarly, the mother's behavior might change as well. Breast milk is actually, it wouldn't match up exactly because the breast milk is mostly fat, I think, fat and sugars. So it doesn't match up perfectly, but it would be really, really interesting to do. And it's something that, I've been speaking to Steve about in terms of a follow-up study.
Thanks, Ola. That was fabulous. With the glucose tolerance, you had that sort of bimodal response. If I remember correctly, the high protein ones were also much lower in lean mass. So does that explain the difference? And did you have different mechanisms underpinning the glucose control at the two extremes?
We have quite a large sample size. We have a very large sample size for that. So what we could do is go back and look at whether lean mass predicts their glucose tolerance. That's a really good idea and something that we can do like straight away, actually. So we can. The glucose levels were given for total body weight, not for total lean mass. So that could potentially be a slight confounder. But I think if we add lean mass as a covariate and see whether that explains it, that could answer that question. So that's a really good idea.
And if I could also ask quickly, I presume that these were, the diets were similar to what we've used here. So they were all defined components. So casein.
Yes.
When you did the experiment where you switched them from the experimental diets to you called it a standard diet.
Yeah, it was.
It
was a standard chow. Actually, it wasn't. It was a standard chow because the diets
so
really
you had a big component switch.
Yeah, so they did have, and I was surprised. I was actually nervous about doing that switch. I was really worried that the mums would just ignore, eat their offspring. But we really didn't have any problems. It was amazing. I think because we did it over Christmas, actually, we had nobody going into the room. Like that all happened over Christmas, which is really fortuitous for us, not for Cam. But that's. Would you be concerned about that? I mean, we didn't lose any offspring, but I see it as actually as potential. We definitely didn't go to the same components we used to
be concerned about it in the sense that it's consistent across all of them, and you're looking for what happened with the initial. But there would be different factors that come into play. So another question that I was going to explore later was, again, with that bimodal one, it struck me that when you look at those defined component diets, it's really only the fat and the protein that will specifically have things that might be essential nutrients in it. The carbs are just sugary crap. And so you could have an effect that some nutrients in those two corners could become limiting in ways that others won't. And then when you go to the standard chow, that effect would disappear a little bit with the new content.
Yeah, I definitely feel like there are limitations with those 10 diets. With those 10 diets, there is kind of covariation between the macronutrients, and I did want to follow up on the first experiment and vary energy levels across it as well. But I haven't been able to get funding to do that experiment. I would like to.
Hi, great talk. I just have a broader question. I was just wondering about if the kind of macronutrient targeting across the non-reproductive phases in life have sort of an impact, and are there differences between if they impact the things. The reason I'm asking this question is because in humans, the taste preferences change significantly during pregnancy and across trimesters. So how do you sort of maintain the isocaloric diet, and if the other phases of life have an impact on a pregnancy or maternal health overall?
So if what they've been eating pre-pregnancy I'm sure they would, because they would probably, over an extended period, they would influence body weight. We know, like I say, we saw in the control animals, once they've been on those diets for 3 months, then we were seeing impaired glucose tolerance. We know from the work here that those diets influence lifespan. So if you put non-reproductive animals on those diets, they strongly influence metabolic health, obesity, and lifespan when you keep them on those diets for 2 to 3 years. So yeah, there would definitely be interactions with other time periods. I thought was there another question?
Yeah. Do you struggle to maintain isocaloric diet during pregnancy and at the lactation phases?
Yeah.
So to eat more, and especially with the changing fat concentration.
Yeah, no, good question. I haven't got the plots here, but we did look at food intake across those reproductive periods. So they do eat differently. The virgins will eat slightly more of the low protein diets. And we saw the same thing during pregnancy. So all of the animals eat more, but the low protein animals eat even more. So they maintain targeting for protein during pregnancy. During lactation, actually it switches. And if I remember correctly, animals who are on the low fat diets ate the most. So they were eating even more. So that does cause changes in energy intake. However, we include food intake as a covariate in our analysis, and that doesn't explain any of the effects. But yeah, there are some really, really interesting changes in terms of food intake and what mothers are presumably targeting and what they need. So they are changing their food intake in relation to those macronutrients differently, particularly in the context of lactation.
Thank you.
Thanks very much, Mike. Really, I also had the same reaction you've heard from the others. Very interesting work. Lots of questions. I'd like to come back to for a moment. That's really striking effect on the junctional zone, which you told us is the primary site of placental hormone growth factor production. So I'm just curious to know whether there are changes in the blood levels, either in the mother or in the fetus, arising from the higher protein diets.
So in terms of placental lactogens, placental hormones.
For example.
So we looked at, I took this one out. This is the one slide I took out because I was worried about time. So this is placental lactogens in pregnancy in response to those diets. So this is basically looking at the kind of lactogenic capacity of the mother's blood. So we did a test where we look at the mother's blood and its ability to activate the prolactin receptor. There are loads of placental lactogens, so it's hard to measure them individually. Actually, we found, found probably not what I would have predicted. I found higher placental lactogens on the low protein diet, and then also going back up on the high protein diet again. This has been reported before, actually. So low protein diets increase the expression of placental lactogens in the placenta during pregnancy. People have looked at like expression of genes. I feel like this might be occurring in response to placental stress. Indeed, when we look at the offspring health, we see it's the low protein and high protein that has a negative effect on offspring health. This might be some way that the placenta is responding to however they perceive that stress in terms of, furthest away from the optimal target of macronutrients they need.
Yeah, I think I'm right in saying that placental lactogen does have an effect to increase insulin resistance, reduce insulin sensitivity. So this runs counter to that notion that...
I mean, they do not. The problem is they do that, but they also drive the pancreas changes. So, like if you knock out the prolactin receptor during pregnancy in the pancreas, you see impaired glucose tolerance. So they're beneficial in terms of actions on the pancreas, but they can slightly impair insulin sensitivity in different tissues. They do a whole load of stuff in the brain. So yeah, it's kind of complicated.
I mean, if you go in the other direction and ask the question whether there are growth factors, local hormones being produced in the junctional zone in response to the high protein that might be driving fetal growth and viability, it also gets very interesting, doesn't it?
Yeah, I would really like to understand what's going on there. Hopefully we will.
So I'll throw in a little bomb at this point. One of the problems with mouse research, as you know, is that there are surprising differences from one strain to the next with some physiological responses. And so I'm just curious to know whether the data you've shown us today holds across different mouse strains.
Wow. I feel like you and I have shared an office for 3 months now. So you're not allowed to review any of my papers going forwards. So you'll never be able to ask that question. You're never allowed to be a reviewer. I mean, quite possibly. And actually what I can say is a whole load of other experiments in our lab are looking at effects of immunological memories of mates on future pregnancies. That's like a whole completely separate question. And we've looked at mating. So we've mated B6 mice with either FVB or BALB-C males, and the placentas are a completely different size, are a completely different, morphology. So absolutely, I mean, I don't know what we'll see. And I think it would probably be quite consistent because at the end of the day, they need the same set of macronutrients. But yeah, we would expect an interaction.
Okay, thank you.
We need to walk.
Do we have lunch today after? Or was
I believe so.
Okay. Yeah.
One more question.
I'll make it a quick one. Thank you for your talk. I was just curious about your glucose tolerance test results. I'm curious if part of the issue is how we measure glucose tolerance, where you're just giving a large amount of glucose at once. So if you have an animal that's adapted to eating high protein, low sugar carbohydrate, and then they're given a lot of sugar at one time and they're less sensitive to the insulin. I guess if you're eating low sugar consistently, does it matter if you have lower glucose tolerance?
I think that's a really, really, really good point. It has, I think Sam Solomon-Beer she actually shared a bit of data in 2 diet comparisons when we first started thinking about that. And that was her thing. It was like she did high protein versus low carbohydrate just as 2 diets. And she said that same thing with carbohydrates, whether when they're encountering a high level of carbohydrates, they have an impaired effect. I'd really like to know if there is a way in mice that we can just like assess circulating glucose levels over a 24-hour period. You know, that would be a really nice thing to be able to do. Like if there's some kind of continuous glucose monitor for a mouse. There is. Well, then we need to do that. I think that would really help kind of show that it's occurring irrespective of a bolus of glucose. So yeah, no, I think it's a good point. It could potentially be a confounder. Yeah.
Okay, thank you so much, Mike. And we can continue these conversations in the kitchen for lunch if you'll join us. So thank you.
Thank you. We wish we'd like. No, we didn't. We didn't see anything like that. I wish we'd looked at a whole load of stuff. When I was down in Adelaide last week at a reproductive biology centre, other people were saying you have to look at the microbiome in mothers as well. So I don't know what you think about that.